Um, okay. Thank you so much. New room already. I hope nobody is in 119. But yeah, this came from the registrar above my pay scale, so has nothing to do with that. Hopefully, will not create too much confusion, and hopefully, there's nobody in 119. Although, Eric, if you don't mind, is there any way you can take a look if there is any student that was confused and is in 119, and then redirect the students here just in case? Thank you so much. Okay, so who has taken classes with flipped classroom format? Okay. Okay, so for those of you who are new, I hope that everyone has watched the pre-recorded lecture before this classroom. And again, the goal of these 80 minutes that we will spend here is not for me to repeat the same that is recorded already, because that will be a waste of time for you, right? Like, because you can watch that on your own. You don't need me. The point here is to a little bit test how much you have absorbed from that, and for you to reinforce by asking any question you have from the material, and then we'll give some time, depending on how it goes, for advanced questions, okay? And I answer as much as I can. Okay, so a little bit of a summary. So in the lecture one, we talk about why we study immunology, and the immune system can influence many different diseases, in good ways, protecting us from infection and cancer, but also causing autoimmunity and allergies. And also some diseases that are not traditionally related with the immune system, neurological diseases, cardiovascular, etc. We talk about the role of the immune system mainly is to protect us from microbes, so they don't cause disease. We have 4 types of microbes that we have to defend against. That is bacterial, fungus, parasite, and viruses. And we also talk about the concept of virulence, being the capacity of the pathogen to cause pathogenicity, to cause disease, right, by damaging the tissues. But that is calibrated by the pathogens so they don't kill their host too quickly before they are able to transmit. And so the pathogens are selected not by their virulence, but they are selected for their capacity to replicate and transmit. And last, we did a little bit of introduction to innate and adaptive immunity. We talk about how innate immunity is rapid, is always ready, is preventing, entering of pathogen. And while adaptive immunity needs the clonal expansion, so it's slow. We talk about innate immunity to have limited specificity. So it recognizes some structures that are present in pathogens, right? But it's like a number of structures that are, defined, right? And instead, the adaptive immunity have these receptors that are rearranged while the cells developed, and they are randomly rearranged. So they generate these infinite numbers of receptors that have infinite number of specificities that can recognize many different structures, right? And we talk about how we can put the fingers in different positions, and that will be random for a particular T cell or B cell that develop in the bone marrow. And so it has billions of specificities in the adaptive immune system. The receptors of innate immune system are encoded in the germline, like all our proteins, right? Versus the receptors of the B cells and T cells that recognize the pathogen structures are rearranged during development. Means that the DNA are constituted by pieces of DNA that randomly come together. and we will learn about that. And so because it's random, your receptors may not be the same as mine, and again, it generates billions of specificities in a single lifetime. The specificity of the innate immune receptors is broad. It means that, for example, I have a receptor, an innate receptor that we now know based on the recent video that is called pathogen recognition receptor, right? Like those are the innate receptors. This will recognize, for example, lipopolysaccharide that is present in all gram-negative bacteria. Okay, it's not present in our cells, but it's present in all gram-negative bacteria. And so it's not that it recognizes one gram-negative bacteria like Salmonella. It recognizes all gram-negative bacteria because it recognizes structures that are in a broad, in a large number of bacteria, okay? And that means broader specificity. One receptor can recognize a large number of bacteria through the binding to LPS. Instead, the adaptive immune system is highly specific, okay? The adaptive immune receptors are highly specific, so they will have a structure, a lock kind of system, just to make an analogy, a lock key kind of system, and that structure needs to fit perfectly, right? And that will recognize a little piece or sequence of amino acids in a particular protein and a particular virus, okay? So one receptor may recognize the amino acid 23 to 33 in the glycoprotein of HIV. And that receptor will not recognize any other thing than HIV and this particular sequence of amino acids in a glycoprotein of HIV. So it's very specific, not only for the virus, but for a particular protein of the virus and for a particular piece of the protein of the virus that is called epitope that, and we will learn that. So it's highly specific. And then we also learned that the innate response is short-term, so basically it doesn't generate memory, but the adaptive immune response can be long-lasting. So it will react upon seeing the pathogen for the first time, that's the primary adaptive immune response, but then it will remember the pathogen and we will learn how. And so the second time it reacts, it will be much efficiently, and that's the memory response or secondary response, okay? Okay, that's the lecture we already went through. We also talk about this graph. If you lack innate immunity, really, because you need innate immunity to trigger adaptive immunity, and we will learn more about that, but the dendritic cells that you already learned about these ones are essential to prime T cells. And T cells help B cells. And so basically you need innate immunity for adaptive immunity. And so if you don't have innate immunity, basically you don't have any of the two. And so that means that the microorganism cannot be controlled at all. If you don't have adaptive immunity, the innate immunity may delay the replication of the organism, but many cases, and depending on the microbe, really, but in many cases will not be able to eradicate the microbe, and so many times the microbe sustain the replication. And if you have both, in this case, with this microbe, it will be contained. Okay, so that's with the lecture one, that's what we reviewed last week, and hopefully Saturday night with popcorns, you watch the video of lecture 2. No, just trying to be funny to wake you up. But basically, this is the summary for the video, right, for the lecture 2. And in this video, we learn generalities of the innate response. Again, review from the first lecture 1, that is rapid, is in all animals and in plants, and has limited specificity, right? And through germline encoded receptors, okay? The fact that it's germline encoded receptors also is the limitation for having, limited specificities, right? Like you cannot encode billions of specificities in your genes. The only reason why the adaptive immune receptors can have billions of specificities is because they have this rearrangement, right? Like they bind one piece of DNA with another one in a random manner, and so there is combinatorial rearrangement of DNA pieces leading to many different transcripts that encodes proteins that are the receptors that have billions of specificity. Okay, but that for the innate immune receptors, they are in germline encoded, and we said they have limited specificity, right? Like we have dozens, would say, of specificities, right? Like for LPS, we talk about double strand, not in this lecture, but LPS is one example, and in the next lecture there will be more. For example, another one, and I'm gonna mention this, I think that I mentioned last lecture too, is double-stranded RNA, right? Like double-stranded RNA is another pump. What is pump? What means pump? Pathogen-associated molecular pattern, exactly. And so that's the ligand of what? That will be recognized by what? The PRR, the pathogen recognition receptors, right? And these are the innate receptors, okay? So in one pair of PRR and pump is toll-like receptor 4 that recognize lipopolysaccharide. Toll-like receptor 4 is the innate receptor, lipopolysaccharide is the pump, okay? Another pair is toll-like receptor 3 that will recognize double-stranded RNA. Double-stranded RNA is another pump because it's generated in the life cycle of all viruses, okay? All viruses at some point generate double-stranded RNA. And so it makes sense that we have evolved means to recognize double-stranded RNA because that means that there is a virus there, okay? And we wanna recognize and alarm and destroy, right, as part of the innate response. Okay, so we also said that there is some intrinsic innate defense in all the cells in our body, okay? And we will see, for example, type 1 interferons are an important cytokine that alarms and destroys as well, and we will learn that. And all the cells of our body can, when they see a virus, when they are infected with a virus, can produce a little bit of type 1 interferon. So why do you think that the cell intrinsic innate response is in all the cells in our body? That all the cells in our body have some innate capacity for innate defense. That means that they will have some PRRs because the innate defense will require recognition of the pathogen through the PRR, right? All starts with recognition. If your body cannot sense that there is a foreign presence, nothing will happen. And that makes sense because we don't want to be just constantly, having an innate response. First, because any immune response will have some a cost and damage of the tissue, right? And second, because it's energetically demanding, right? So we only want to induce an immune response when there is a pathogen. So all starts with the sensing. And so, again, we said all cells in our body have this intrinsic innate response, and that starts with sensing, and that starts with pathogen recognition receptors. And so why do you think that all the cells in our body will have the capacity to have some degree of innate, cell intrinsic, cell autonomous, innate response. Okay, I'm gonna do a confession here. I am truly as innate immune system, okay? I have no memory. So I will not remember anything at your faces if you raise your hands and say something that, don't feel that you are being evaluated here at all, okay? And so you can say whatever thought you have, and that will trigger, somebody else's thought, and it will help your learning as well, because when you participate, you can fix more. I have my son is now in college in sophomore, and I try to tell him, and I don't think I have succeeded, but I told him, raise your hand and ask questions. College is perfect because nobody expects that so you can ask any question. That's fine. That's why you're here. So anyway, let's see if I can succeed with you. Anyway, so why do you think that all the cells in our body have cell intrinsic innate response? Yes?
To ensure that the pathogen is detected as quickly as possible to get that response started?
Absolutely. Timing, excellent, right? And also, pathogens evolve. They can infect any cells, right? Like we have microbes that have, we don't even know they will emerge, right? And they could infect any cell in our body. And so it makes sense that all cells have some level of protection. And then timing is also important, because if we have innate response in all the cells, it means that the first cell that was infected, right, will already sense an alarm, okay? And so recognize an alarm. And so that is saving time. It's already quickly, we don't need the pathogen to spread and infect innate cells and the immune cells to sense. We can have any cell in our body to start this to some degree. And epithelial cells are an example. They are not traditional immune cells, but they have pathogen recognition receptors. They can recognize and they can initiate alarm through cytokines and chemokines and attract immune cells. And then these are more the professional innate cells, right, that are part of the innate immune system, that are neutrophils, macrophages, and dendritic cells and natural killer cells. And there are other cells that are part of the innate immune system that are basophils, eosinophils, and mast cells that are also involved in allergies, and so we will learn them much later. And they are also important for defending against helminths, okay? And we will learn about them later. The neutrophils and macrophage, we learn that they are important for phagocytosis. They have reactive oxygen species. The neutrophils particularly are called these walking bombs, right? Because they go to are the quickest one to get to the site of infection. And they will get there and they will phagocytize, but also they will release their content through netosis, N-E-T-O-S-I-S, netosis. And basically this is a mechanism that was relatively recently discovered, in which the neutrophils release their chromatin like a net, and through that they trap extracellular bacteria. So the macrophages also are very professional phagocytic cells, and they also release inflammatory factors, and so they contribute to inflammation, and we will, I guess that that was also covered in the lecture. We will talk a little bit about that. And then let's talk about inflammation. So macrophages, dendritic cells, and also natural killer cells to some extent, they release, but mostly macrophages and dendritic cells release inflammatory factors, and this cause inflammation. And we talk about inflammation, you know, even in the TV, in the commercials, right? Inflammation. And so what is inflammation, right? What is inflammation? How it starts, okay? Because there is so much money now put from biotechs, right, in trying to solve this chronic inflammatory disease. And this inflammation has been associated, for example, of course, with autoimmunity and arthritis and, you know, the traditional immune cells, but also with cardiovascular diseases, with Alzheimer's, for example. And so there is so much interest in treating chronic inflammation and curtailing this chronic inflammation and how to resolve inflammation in a manner that doesn't destroy the tissue, for example. And so what is inflammation and how it starts? Who wants to throw the first? What is the starting point for inflammation? Close, very close. Excellent. So complement pathways can trigger inflammation. So you are, you know, very close. But there is a general event that causes inflammation, right? Yeah, in the back. Increase in vascular permeability, right? That's the whatever triggers this increase in vascular permeability will cause inflammation, and that's because it like complement factors like C3A and C5A, they cause and TNF alpha, that is a cytokine that we will learn as well. They cause dilation of the vessels. So imagine you have a tube and then you need to make it wider, right? It dilates, and now the cells that were the endothelial cells, because it needs to be wider, the endothelial cells start separating, and that causes extravasation, right? Cause increase in vascular permeability. Okay, so this dilation, okay, that leads to the increased vascular permeability that is called, for example, by C3A and C5A, that are called also anaphylotoxin. These C3A and C5A are called anaphylotoxin. So, but this dilation that caused the increased vascular permeability, also this dilation will have result in more blood getting to this area, because now you have bigger vessels, right? More dilated vessels. And so there will be more blood getting into this area where C3A and C5A were released, correct? And of course they were released because complement was activated. And of course complement was activated because there was a pathogen there, okay? So it makes sense. And so now if we have more blood getting to these vessels that are dilated, what would be the consequence of that in terms of inflammation characteristics? Yes. I would say redness there, okay? You have more blood and you will have redness. But swelling is another characteristic of inflammation. And that directly, the direct cause of that is that when you have dilation, you have more space in between endothelial cells, right? Because they are separated. And that's when the plasma and proteins will leak out of the vessels. And also the neutrophils, the leukocytes, will also extravasate. And so this accumulation of liquid and cells will lead to the swollen. Excellent. Very good. And what is another characteristic? Yes. Heat. And why is the heat? Yes. The blood is warm, but also there is more blood there, more blood circulation, and that increases the heat. And then the other one is the pain, right? That's why we don't like inflammation, because it's painful, right? When you have an inflammation in the knee, it can be paralyzing. And so the pain. And why is the pain? Yes.
The increased amount of blood cells and stuff in that area causes pressure on nerves.
Exactly. So it's the pressure on the nerve endings in the area where there is accumulation of cells and liquid, and that causes the pain. And so then these are the four, you guys went through the four characteristics of inflammation, right? Redness, swollen, pain, and heat, okay? Calor, rubor, you don't need to learn them in Spanish, but yeah, that's it's calor, rubor, dolor, and I'm missing one. Tumor, which is swollen. Okay, good. So the macrophages will contribute to, and the dendritic cells will contribute to inflammation because they release cytokines, right? And that a major one there is TNF-alpha, and we will learn about this one. And that TNF-alpha promote a cell dilation and extra increased vascular permeability, and that's how it's initiating the inflammation. So neutrophils and macrophages are like really good phagocytic cells, and then dendritic cells are excellent links, and are essential, are the bridge between innate and adaptive immunity. In our lab, we work with dendritic cells, so I'm very fascinated by these cells. And I love them because they participate in innate responses, they release cytokines, and they help contain the pathogen, right? But also, it's super interesting because they will get information from the pathogen, and we don't understand everything. But they get information from the pathogen through their pathogen recognition receptors. That's the only way they can get information, through sensing, right? And they get that information, and they communicate this information to the T cells. And without dendritic cells, you cannot have activation of the T cells. So now they are telling the T cells, okay, this is the pathogen, and this is the type of response you should be mounting for fighting best this pathogen, okay? So if the T cells are the soldiers, the dendritic cells are the generals. They will tell the T cells what to do. And then the other adaptive immune cells, the B cells, they need, for the most part, they need T cell help. So if you don't have dendritic cells, then really you will not have adaptive immune response, because they are key for initiating T cell activation or T cell priming. That is another terminology, okay? Okay. And then we have natural killer cells that, as the name indicates, are cells that can induce cytotoxicity and apoptosis on the infected cells. And we will learn about these ones a little bit more in next lecture. Okay, how does the innate immune system work? We said it will be preventing, and that's all the time, right? And then we learn in this lecture that prevention can be through physical or mechanical barriers, like the epithelial cells that are in our skin, but also in the gastrointestinal tract, urogenital tract, etc. It can be through chemical barriers. Another physical barrier is the fluids, and like perspiration in the spleen, and also mucus in gastrointestinal tract, for example. Okay, the other barrier, the other mechanism for preventing infection is chemical barrier, and that's through release of enzymes like lysozyme that will degrade the pathogen proteins, and also through low pH that will diminish the or disfavor pathogen replication. And what other chemical barrier? Remind me. Yes. True that. Microbial peptide. So these microbial peptides will form pores. They act in different ways, but one way is forming pores in the a wall of bacteria and then leading to destroying the pathogen. And then we have the microbiological barrier. And so what do you know about that? Okay, so yeah, let me see. What do you know about microbiological barriers? Yes.
There's like groups of commensal bacteria.
Yes, correct. Yes, commensal bacteria, and these commensal bacteria are with us all the time. They are not only in the gut, but they are also in the skin. They are in the respiratory tract, and so they will. They are great because they synergize with us in many ways. They help us process the nutrients, but also they compete with pathogenic bacteria, right? And so many pathogenic bacteria are successful because they are able to outcompete and, for example, deplete of nutrients the commensal bacteria. And so they establish a niche for them to replicate best. And so I really encourage you. Have any of you watched these global immunotalks that I recommended in the video? I know, it's a lot. And so, you already need to watch the video, which is mandatory and is priority, and, having this extra. But I do recommend you to watch these ones. The two that I recommended, they are, as I said, one is from a colleague of mine at UCSD, Manuela Rafatello, that works on microbiome and how bacteria influence many aspects, or commensal bacteria influence many aspects of host physiology, definitely immune responses. If you search global immunotalks and my name, I also gave a global immunotalks in which part of the talk involves a commensal bacteria that is Akkermansia mucinifila that we show that goes up when there is a fasting. Any of you is doing the fasting diet? Oh, you're too young, you don't need to do it. but when you fast for 2 hours, for 2 days, Akkermansia mucinifila would go up, possible even less, but also when there is anorexia due to an infection, Akkermansia mucinifila will go out. And we show in a particular setting how this bacteria is able to modulate immune responses in the context of a viral infection. So I recommend that global immunotalk as well. So if you go globalimmunotalk.org, you will see all the global immunotalks, and you can also Google global immunotalk Zuniga, and it should be. And if you need, I can also send the link to the video. But anyway, so it's fascinating. And, I'm sure you learn also, if you took microbiology, to talk about this, what used to be called flora, we don't call that anymore, but this microbiological barrier. Okay. So that's preventing the entry of the pathogen. We have also the recognizing the microbe, and we already talked about complement. Complement is a set of proteins that are produced by the liver, are serine proteases, and it's a protease cascade, how it acts. And basically, it's beautiful to review all the three parts of the innate responses: recognition, alarming, and distraction, right? Because complement can be activated in 3 ways. Which ways? Classic, alternative, and lectin, right? And which is the first one that is activated? Alternative. And that's activated because of some complicated chemistry that I wouldn't be able to teach, in which the pathogen surface creates an environment that activates complement. And then we have the lectin is activated through lectin receptor. And then we have the classica that is activated by antibodies. So it's only activated later when the antibodies that are derived from B cells from the adaptive immune response are already secreted, right? And so the classic, that's why the classic is the latest one. And they all converge in which step? One central step. They all lead to cleavage, yes, of this C3 complement factor. And it will be cleaved in two pieces that are called C3a and C3b. And so C3b will induce opsonization. What is opsonization? Exactly. So marking a pathogen to enhance its uptake. That's opsonization. And it's not only made by C3b. C3b is the first opsoning we are learning, but there are other ones, okay? Other ways to opsonize a pathogen. But so that will lead to opsonization and will also lead to cleavage of which complement factor? C5, in C5a and C5b. And then C5a together with C3a, and we were just talking about that, are the two anaphylotoxins that basically what they will do is increase vascular permeability, induce vessel dilation. And when you induce vessel dilation, what do you have? Inflammation. And they also are chemotactic, and so they will help recruit immune cells to the site of inflammation, okay? And so that way they are alarming. While C3b, that induce phagocytosis, that's one way in which it will be destroying the pathogen. That's one way in which destruction of the pathogen occurs, is by uptaking this pathogen inside phagocytic cells. So the phagocytic cells, like macrophages or neutrophils, now destroy the pathogens through these reactive oxygen species that they have in their vesicles. Okay, and then we have C5b, right, that it will initiate the formation of the pore in the membrane of the pathogen, and that is another way the pathogen will be destroyed by these complexes that are forming of proteins that are forming a pore. So basically the pathogen wall is destroyed, and that's how the pathogen will be killed. Okay, so that's in a nutshell complement. And these are basically soluble proteins, right? Like where is complement? Where can you find the complement proteins? Plasma, excellent. Are there in the tissues? Like, let's say you have the skin, and then underneath the epithelial cells that we were just saying is part of the physical barriers, you have mucosal tissue, right? Cells of the tissue. Do you expect finding complement there all the time? One yes? How many yes? How many no's? It needs to add up, it's yes or no. Okay, the answer is yes. Yes, you have complements in the tissues as well. It's not activated unless you have a pathogen, but it's there. And then why it makes sense that it's there underneath the epithelial cells? Why you would like to have complement there? Because if you have a cut, right, and so you don't have any more of this physical barrier, and the microbe gets into the tissue, and now the complement can be activated quickly through the alternative pathway and initiate inflammation, and so basically recognize, alarm, to get more help and start destroying the pathogen as well. Make sense? Okay, cool. So these are soluble proteins that are in the blood, in the plasma, in the lymph, and in extracellular fluid that is in the tissues. And we also have cell-associated receptors, okay? And so these are pathogen recognition receptors, such as toll-like receptor, and I will look into these ones more carefully in the next lecture, but some of them, the classical ones, are toll-like receptors that are in the membranes, that are associated in the membranes, and we have other ones that are in the cytosol. Okay, they are not membrane associated, like Rig-like receptors or SIGAs, and we will look into these ones. And so these cell-associated receptors can be in the membrane associated in the surface or in the vesicle membranes, or another group can be in the cytosol. But all these cell-associated receptors, these are called pathogen recognition receptors that will recognize PAMs, which are pathogen-associated molecular patterns like LPS or double-stranded RNA, okay? These are structures in the pathogens that are conserved in a group of pathogens and that we don't have, okay? And these pathogen recognition receptors that are the ones that will recognize the PAMs are, once they bind to the PAM through an adaptor protein that varies depending on the pathogen recognition receptors, will trigger a signaling cascade that will lead to the activation of a transcription factor that will bind to the promoters of several genes that will then be transcribed. You will have proteins that will be aimed. These proteins that are produced in response to the activation and recognition of the PAMs will be tailored to do what? What do you want to do if you were an immune system, an immune cell? What do you want to do when you sense the presence of the pathogen? Just whatever makes sense. Destroy, and what else? What else? If you see an invader in your home, right, you want to destroy the invader and call for help, right, alarming the rest of the neighborhood, right? And so these genes, when the pathogen recognition receptor binds the PAMs, the genes that are induced, right, and the proteins that are synthesized in response to the recognition of the PAMs are proteins that will be aiming to alarming and destroying. Okay, and it's funny because we don't know all, we know there are, hundreds of genes that are downstream of pathogen recognition receptors, and we don't know the function of all of them. But we surmised, right, that as many of the ones that we know the function of, this must have a role in alarming and destroying, okay? And then this is a little bit more advanced, so I don't want you to get confused. But there is another thing that makes sense that you would like to do when you see this. And this is way more advanced, so I'm hesitant to mention or not, but let's see. What do you think? What else you may want to do that when you have an immune response? You definitely alarming to get help and destroying, right, the pathogen. But we also said that any response would lead to some damage, right, of the tissue. So, yes?
To try to coexist with pathogens.
Excellent. Yeah, in a way, kind of, sometimes you may wish to coexist, avoid excessive immune response. And so you would also trigger some immunoregulatory mechanisms that will attenuate a little bit the immune response, so you don't have excessive immune response and death due to immunopathology. Excellent. And so that may lead to coexistence with the pathogens, Or maybe, depending on the pathogen, how the dose, the route, it may be, you attenuate the immune response, but you still eliminate the pathogen, but you attenuate the damage in the tissue. But excellent. There will always be some damage, right? And so if there is always some damage in your house, what do you want to do if there is destruction of the house? Yes. Excellent, excellent. I love how you guys are thinking. Limit the distraction, which is similar, right? Like some inhibitor. At the same time, you will not try to, you will try to minimize the distraction and still, contain the invader, right? But let's say, and I agree with that, right? Like that was the previous point. But there will always be some distraction, right? If you have somebody trying to rob your house. And so if there is some distraction, if something is broken, what do you have to do? Repair, right? You will need to start, and that may come with a delayed kinetic, but that's another thing, and that's a little bit more advanced. These 2 points are more advanced, so you guys are thinking really well. Because there is also repairing, and there is more and more emphasis in how, innate receptors, and they will also trigger some repairing mechanisms. And there is a major role of immunity in wound healing, okay? And so that's interesting as well. And again, it's a little bit more advanced. We're not going to be evaluated. I just love, that to make a bigger sense of it, and I want to share that with you, and you basically are figuring it out on your own. Okay, so what we will do, is there any question? Yes?
So does the PRR bind the PAMP and then that complex binds the adapter, or does the adapter help the PRR bind the PAMP?
Excellent. So the PRR combine the pumps without the adapter. It's like an interaction that happens. But now the adapter is always associated with the PRR. It's always associated. And when the pump binds the PRR, it triggers some, and the mechanism changes depending on the PRR. Many times it's a conformational change or phosphorylation and of the adapter. And then that leads to the signaling event. Mm-hmm. So it depends, the modification of the adapter. The adapter will help the signaling, right? It's like a bridge between the PRR and the starting of the signaling. That can be kinases or some cases proteases, but mostly kinase, yeah. Okay, any other question? Okay, we have some time, so let's start with some, clicker questions, but we are not evaluating clicker today, so we just do the clicker for, just a test for, let's see if we can do it, but my goal is, that we will start clickers in the 3rd week, so next week, okay? So we have 2 days to practice to make sure everything is okay. Or wait, we said 3rd lecture. I'm confused now. 3rd lecture, which is Thursday. Okay, sorry. So 3rd lecture, so Thursday, we'll start counting the clicker, okay? Let me see if I can connect this because there is no adapter here. So we are missing an adapter in this room. Okay. Let me see. Okay, let's do this. Okay, so give me a few minutes, and you guys have the slides for the flipped classroom, correct? Right? And so we start answering on your own the clicker questions, and then we will do them together, okay? But we will go fast afterwards because you are starting right now to look and mark whatever you think that you are correct. And we will, yeah. Yes, so if you go to Canvas, modules, and then you have syllabus, lectures, and there used to be flipped classroom slides. They are. Oh, thanks God that you guys told me. Huh. Okay. That's. And you didn't see the slides for the lecture either? Ah, okay. Okay, so let me do this right now. And so, yeah, great. Okay. It's here. Maybe it wasn't published, you know. Huh. Flip classroom slides. You don't see them. Ah, okay. Okay. Okay, there you have the first one. Yeah. So it should be there now. Okay, I will take care of the rest, but now I want to make sure. Anyone has a connector from a flash drive to the thin ones or HDMI? Yeah, you do. Because that will allow us to practice with the clicker pace. Yes. Oh gosh. Thank you so much. Ah, no. It's HDMI. It needs to be this flash drive for the clicker. Oh, yes, yes, yes. Thank you. Okay, you guys start. Everyone found it in a Canvas? Okay, so you start marking your correct answer. You can talk to your neighbor if you wish, or you can do it on your own. Start doing that because we will go quick afterwards, okay? Okay, guys, I think I can pull this. Let's see. Okay. Okay. So you guys wanna check if you can connect and we can start. I will start. I think I need to press pull and start multiple choice. Oh, wait. Okay. Again, I think that this is the pool. This is where you need to join, and then we will do this question first, but I just want to make sure everyone can join. The frequency here is AC for this base, and this is the link. I think the code, I don't know if your clicker is asking the code, is this QVDW. You couldn't find this because it says frequency AC. And I can see some numbers coming already, some answering, no? Who can get the answers? Who can, yeah? Huh. Say again?
I just have the app.
And with the app, can you do it? Okay, can you do it with your clicker, the physical clicker? No. No AC? Yes. It worked? Okay, can you try AA? No? Yeah, it seems that it's working. Okay, thank you so much. Whatever, it works. Okay, so let's do them quickly. How many did you get so far? How many clicker questions were you able to answer in this time? All of them? You guys are cracks. Okay, excellent. Okay, let's do them quickly then, and I want to make sure that you understand and there's no question, okay? Okay, the immune system adverse reactions can contribute to which of the following conditions? Neurological diseases, diabetes, heart disease, arthritis, all of the above. And I think that, let me see, I can go here and I can stop. And I will always make sure that you have, it's very difficult for me to do it on the screen, so let me. Mm-hmm. I cannot do it. I need to grab this bar and take it to my computer, to my screen here. 1 second. Perfect. Okay, so I will always tell you when I would stop the poll, I will say, now make sure you vote, because remember you get points and it counts even if you answer the wrong question, the wrong answer, okay? So we will stop it now. So make sure everyone voted. And so it seems that we are in this one. And it seems that almost all of you answered the correct one, which is all of the above. And I can show you this here. I know I can show. Okay, sorry for the technical thing. It's the first one, and it should work the next one. Okay. Anyway, I'll show you the next one, but almost all of you answered. It's quite easy, right? Like we talk about this being non-typical immune diseases. But that's huge, right? I mean, that means that if you can manipulate the immune system, and that's why we work super hard as scientists to understand the immune system, because it can have a lot of impact. in many of diseases that we care about. So, and you know, if you're here and you learn and you, hopefully I can engage and ignite your passion, maybe you will try to study the immune system, and then you will be able to manipulate a number of diseases, not almost every disease, I would say, to some extent. Okay, so one reason that pathogenic microorganisms have an advantage in the host they infect is because, and then we will start the poll there. So vote 30 seconds, because you already did it. Okay, I'm about to stop the poll right now. Make sure you vote. Okay, I'll stop it. And so most of you answer B. And I want to show you, and each time I try to show, I need to figure out how to show you. But anyway, most of you answer B, or all of you. And so one reason pathogenic microorganisms have an advantage in the host they infect is because they have previously been encountered through natural exposure. No, not necessarily, right? Reproduce and evolve more rapidly than the host can eliminate them. Strengthen the host that's correct, right? Like we said, the organisms have the upper hand. They strengthen the host immune response. That may be true, but it's not how they have an advantage. Have previously been encountered through vaccination. That's also possibly true, but that's not why they would have an advantage. Are selected to kill the host, and we said that's not true. They are selected for replication and transmission. So let's do the next one. And so I put the poll here. 20 seconds, please, because again, you have already had time to do this. And I can see already that most of you are answering right. Okay, I'm about to stop this and make sure you vote. It's stop. And again, I cannot show it. I'm doing something wrong. Which cells is the origin of all blood cells, right? Like we say, all blood cells, hematopoietic stem cells. And we saw how this can give rise to the common myeloid and common lymphoid progenitors, and then each of them will generate different immune cells. The common erythroid progenitor, megakaryocyte progenitor, also derived from hematopoietic stem cells and will give rise to the blood cells and the platelets, okay? Okay, next one, and then let's do answer true is A, B is false, and then let's do 10 seconds for these ones, and let's see if this works. Animals, but not plants, have an innate immune system. And this is false, as most of you answer. And let's do the next one here, 10 seconds. Innate immunity is manifest only in hematopoietic cells of the body. And you guys are learning because all of you are answering, or most of you are answering false, and this is false. And so let's do the next one. So we talk about every cell in the body has some degree of innate immunity. Okay, innate response are immediate, rapid responses, 10 seconds. We talk about that. I know that you know that. And then 10 seconds. And again, most of you are answering the correct answer. I'll stop it. This is true, right? Most of you are answering true. Innate response are immediate, are quick, and that's the advantage they have respect to adaptive immunity. So it's kind of the holders, right? Like that will until the adaptive immunity is induced. Innate receptors have broad specificity, recognize elements present in different classes of pathogen, and let's do that one. This is true, have broader specificity, elements present in different classes of pathogen. We talk about LPS, double-stranded RNA, and we will learn more in the next lecture. So I'm going to stop this one. And then the next one, the specificity of innate pathogen receptors is determined by gene rearrangement during development of innate cells in the bone marrow. Let's see what you answer. You can start answering that one. 10 seconds, and I'm going to stop it now. Okay. Oh, look at you. This is easy. Anyway, so it was half and half. And so I think that is because, and I truly think that this is because you didn't read carefully. And that's very important, because many times, and I tell my son this as well, many times you know the answer, you just rush the reading. And so the specificity of innate pathogen receptors, these are the PRRs, pathogen recognition receptor, is determined by gene rearrangement. Gene rearrangement is this cut and paste of different pieces of gene, okay? The pathogen recognition receptors are generated like that or are encoded in germline. Okay, and so let's do this one again. So I want you all to vote again if this is true or false. True is A, false is B. Okay, everyone is answering B right now. Thank you. Okay, so this is false, right? Because the pathogen recognition receptors are encoded in germline. Okay, next one. NK cells arise from which progenitors? And we said common lymphoid progenitors, okay? We will skip this one because it's very easy. In individuals lacking innate immunity only, okay? What happens to the number of microorganisms during infection in the typical case? Okay, in the typical case, that is the one that we studied. So let's go with that one. Okay, and so let me stop it now, make sure you voted. So the number remains constant, no. The number decreases rapidly, no, because it doesn't have innate immunity. The number increases rapidly and continuously. And the reason, that's correct. So C is correct, most of you answer correct. And the reason is because you need innate immunity for adaptive immunity as well. So you will not be able to contain the pathogen and you will not be able to trigger adaptive immunity. So next one, like starting this right now for you to answer. And this is very easy, so I'm giving you 10 seconds, and I'm going to stop it now. So which cells orchestrate the adaptive immune response? And the answer is B cells and T cells, okay? So you know that already. I can go slower, and if you have any question, please raise your hand. Yes, please. Oh, good point. I can see how it can be confusing. I think the question should say which cells belong or mount the adaptive immune response, maybe not orchestrate. Yeah, that's a good point. I can see. The other thing that YC is not correct because it says dendritic cells and NK cells, right? But if it said dendritic cells, I can see how it can be confusing, because it's true that dendritic cells also orchestrate, right? Are the generals. So good, excellent point. Thank you so much. So probably the wording should be which cells mount an adaptive immune response would be much better, and that will be undoubtedly B cells and T lymphocytes. Yeah, thank you for thinking. Yeah, yes.
I have a question about the previous question.
Of course.
How come, or is it that at a certain point you would sort of hit the carrying capacity of the body and you would run out of nutrients and then it would sort of plateau, but it just increases a lot, or do they really just grow continuously?
Oh, excellent. I think that when you the question is how the pathogen can grow continuously. At some point, there will not be any nutrients, right, for fueling the pathogen metabolism. But when that happens, you will be dead. No, but excellent. I love how you're thinking. And so basically, continuously throughout the lifespan of the host. Yeah, excellent. Okay, how does the adaptive immune system respond to pathogen? Let's see how you answer that one. And so 15 seconds since you have looked into that before. Okay, so I'll stop it now. And so the answer for that one is A, by selecting and expanding the right cell from a pool of many lymphocytes. And so we did the log key, remember, analogy, right? Like you all were lymphocytes. And so, but only you has the log that will match the key in the pathogen, okay? And that lymphocyte is now copied, which is clonally expanding. So by selecting and expanding the right cell from a pool of many lymphocytes. We will learn more about this for sure, okay? But just to start introducing how it acts. So the next one is, let's see which one is mismatched there, if you can vote. Okay, this is easy. I'm gonna stop it now. And so none of the above is mismatched. Most of you answer correctly. So chemicals are antimicrobial peptides, but also remember lysozyme and low pH. Okay, mechanical, mucus in the intestinal tract, that's correct. Also remember mechanical are the epithelial cells and also fluid, like perspiration in the skin. Microbiological are the microbiota. Okay, next one. And I'm letting you answering that one. Okay, so I'm stopping this one right now. So most of you answer C. What is the primary mechanical defense of the skin against pathogen? Flow of fluid, perspiration, and sloughing off of the skin. We haven't mentioned this one, but that's true. That's another way, the skin loses the cells that are infected, right? Okay. Any question? Okay. So next one, let's have you answer in 10 seconds. Okay, I'll stop this one right now. So most of you answer B, which is correct. Which of the following is a chemical variant found in the gastrointestinal tract? Acidity and proteases like lysozyme, that's the chemical barrier. That's correct. We'll do the next one and let you answer. Again, I'm going fast because you have had time to answer them. Otherwise, I would go slower. And I can go answer any question. So raise your hand if you have a question. So in the microbiological barrier in the urogenital tract, I'm going to stop this now. And the normal flora, so that's the microbiota, is in the urogenital tract. That's the microbiological barrier, and that's the D as in David is the correct answer. So next one, where are the proteins of the complement system produced? 10 seconds. And they are produced, as you all are answering, or almost all, they are produced in the liver. So we'll stop it now. And most of you answer liver. Let's do the next one. Which of the following pathways is the first to act in complement activation? We'll skip this one because we talk about this one and it's the alternative pathway, correct? C is the correct answer. And so let's see if we can finalize all of them. What is, and I will stop with the clicker so we can finish. What is the role of C3b in the complement system? It recruits inflammatory cells. No, this is C3a. It opsomizes pathogens. This is correct, facilitating uptake and killing by phagocytes. So that's correct. So B is the correct answer there. Okay, so I don't think we will finish all of them, but we will continue on Thursday, and whatever is left can be done in the discussion section. Thank you so much, and make sure you bring the clicker in. We will start counting on Thursday, okay? Thank you, guys. Let me return this to the...